consistent development is required to combat such conditions in the near future. In this context, we hope today's webinar will definitely enrich the audience. The speaker today is Dr. Sundi Panchakabhati. He is an assistant professor from Amity Institute of Biotechnology, Amity University, Kolkata. I, Dr. Chittabrata Mal, assistant professor of this department, am going to moderating this webinar. We have panelists for this webinar, Dr. Somen Kumar Pati, HOD and associate professor, Department of Bioinformatics, Dr. Hidaya Ranjan Boiraggo, assistant professor, Department of Bioinformatics, Dr. Sarojit Das, assistant professor, Department of Biotechnology. Dr. Somen Kumar Pati has taken the charge of our department the day before yesterday, so he could not modify and circular the flyer within this short time. Kindly excuse us for that. So we are running different programs in our department: BSc 3 years course, MSc and MTech 2 years course, and we have different special programs like genomics and proteomics, NGS technology, and many others. The students can join this department if you want to know from very basic to advanced levels of bioinformatics. Next, we have some announcement. The webinar will be recorded and it will be streaming in YouTube. You can directly type questions in the chat box and that will be addressed at the end of this webinar. At the end of this webinar, you will be given a feedback link and a certificate will be issued accordingly. Now, on behalf of our department, I welcome Professor Soikat Moitro, Honorable Vice Chancellor, Maulana Abul Kalam Azad University of Technology, West Bengal. With his constant support, we are able to organize this webinar within a short period of time. Besides his busy schedule, he is present here today. We always love to hear from our dynamic, enthusiastic, and positive-minded VC sir. I am requesting sir to say a few words to this audience. Sir, please.
Thank you very much. Department of Bioinformatics is organizing a very important and relevant webinar, basically on the SARS-CoV2 virus and this evolution of spike protein, and where there's lots of analysis and informatics can be done in the field of bioinformatics to go for predictability mode, to go for this better understanding how this virus can affect this human body. So it is important in this sense. Today I widely came to know from a report that this COVID problem or COVID virus related problem that may continue as long as till 2035. So it means that we will have numbers of these case studies, numbers of data, numbers of this mutant virus, all these things. So and that means huge volume of data will be at our hand. Previously, we did not have, even few years before, we did not have the facility to go for handling big volume of data as per our these tools and techniques were concerned. But because of this emergence of big data analytics and Hadoop platforms and other platforms where huge amount of data can be handled, processed in a meaningful manner, so it is giving us lots of opportunity in understanding these many new things which were restricted or confined only with the study with the limited volume of data. And in many cases, I've observed that this outcome of the study that may not have that much of practical applications or reality. But as because we are now going to handle a big volume of data with different this variety, veracity, and different formats. So now meaningful information and this can be possible from analysis and handling all this data. Bioinformatics department that can play a leading role as far as there is COVID virus related research is concerned in coming decades from our university. And for that purpose, we set up this department long time before, but till now, it has not yet geared up to take up this challenging situation or challenging this scenario as far as the biological research is concerned, informatics is concerned. But I believe that with the proper collaboration or networking with different universities and institutes and different departments and with different experts in this field, now we can do many things. And here today, I'm very happy to say from Amity University, Dr. Sundi Panchakabhati has joined. So this platform can open up an opportunity for our collaborations of research activities between these two universities, McCourt and Amity University, as far as this bioinformatics department or biological research is concerned. And more and more these types of integration is required. And we would like to invite numbers of experts and professionals in this field at our platforms to share their knowledge and to explore the possibility of networking and collaboration in the field of academics and research. Because in today's world, until and unless we are not going for massive integration, networking, and collaboration, ultimately the outcome of all this research and activities that may not serve the purpose of the society, or maybe for a limited period of time it may be successful, useful. In future complex situation, all these studies and this outcome of this works that may not be that much relevant. So this is an opportunity, and another important aspects of this new digital culture is we can use our available time to the best possible extent by organizing these sorts of webinars, technical talks, and meets through digital this video conferencing, digital platforms and meets. And it is giving us enough opportunities to use our time in a more prudent manner. That's why at weekends, I can see that numbers of participants, 271 participants, they have joined this webinar platform to gain knowledge and understanding and to validate their existing knowledge from the deliberation of the experts. So this is a new culture. This culture we would like to, develop further, and we would like to integrate all our learning community through this digital platform in the best possible way. We have opened up our campus for physical, these classrooms and all these things, but that is not sufficient enough to attain a high level of quality within shortest possible time until and unless we engage ourselves in using productively the available time at weekends and holidays by organizing these webinars through digital meets or any technical talks in the best possible way. So I'm very happy that Department of Bioinformatics, they have organized this webinar. My sincerest thanks to Dr. Sundi Panchakabhati for coming to this virtual platform, for sharing his expertise and knowledge with our participants. I believe all the participants, panelists who are present over here, they will be engaged in meaningful interactions and discussions to understand the subjects in a better manner and to find out the avenue to work together in a more productive manner to design and formulate new areas of research. So this can be one of the outcome of these webinars. My best wishes to all the learning community and participants who are present over here, Dr. Chittapoto Mal, Dr. Somen Pati, Dr. Hriday Boiragi, and others. I cannot see Sarojit Das, probably who will be joining later, but my best wishes to all of you, and kindly continue this practice in involving many departments, many experts, many professionals, so that within the shortest possible time, because we have already lost these valuable times, and world is moving fast. So we have to now run. We have to run at a high speed. Otherwise, we will not be able to catch up. So my best wishes to all of you, and I strongly believe that you have the potential and intention to catch up the level of advancement which has happened in the field of bioinformatics in the coming days. So with this, I'd like to conclude. Thank you very much for inviting me and for allowing me to share my views. Thank you.
Thank you, sir, for inaugurating this webinar, and thank you very much, sir, for your encouraging words. We will definitely follow your advice and will try to help the students to reach their goal. we got a huge response from different corners of India and abroad for this interesting webinar, and we received more than 1,000 registrations so far. Next, I would like to invite the panelist, Dr. Somen Kumar Pati. He is the Associate Professor of our department and expert in machine learning and artificial intelligence. Sir, requesting you to kindly say a few words.
A very good morning to all. Thank you very much, Dr. Chittabato, the coordinator of this webinar. We are extremely honored to have with us our Honorable Vice Chancellor, Sir, Professor Soikat Moitro, gracing the inaugural program of this webinar. Thank you very much, sir, for your ever encouraging and motivational speeches. The participants will surely gather an encouraging experience from your advice, and more particularly from the knowledge they received from this webinar. Thank you once again, sir, for your kind presence today. I am very thankful to our participant and ensure you must gather valuable knowledge from this webinar to enrich your future plan and research interest. Hope you will enjoy today's webinar. At last, I am very honored to our respected resource person, Dr. Sundi Panchakabhati, as the speaker of this webinar on the very recent and novel related topic of the bioinformatics. Thank you very much, Dr. Chakraborty. Now I want to hand over the charge to Dr. Chittapato. Carry on. Best of luck.
Thank you, sir. Thank you for your valuable and supporting words. Next, Dr. Hidaya Ranjan Boiraggo, who is also Assistant Professor of our department and expert in structural bioinformatics. Sir, if you kindly say a few words.
Okay. Thank you, Dr. Chittabrata. First of all, we are very grateful to our Honorable Vice Chancellor, sir, for encouraging and supporting us to arrange this webinar. We are also welcome to our respected speaker, to Dr. Sundi Panchakabhati, sir, for providing this scientific talk on SARS-CoV2 or COVID disease. We do believe the computational work of bioinformatics related to COVID disease will definitely provide some new insight about the viral mechanism, and it will definitely help the experimental scientists. Our department, basically Department of Bioinformatics, is working in different areas of bioinformatics areas like NGS, metagenomics, structural bioinformatics, computational drug design, and artificial intelligence also. I hope all the participants may definitely get some good knowledge from this webinar. Okay, thank you, Dr. Chittapato.
Thank you, sir, for your valuable words. Now, with your permission, I would take pleasure to introduce today's speaker of this webinar, Dr. Sundi Panchakabhati. Dr. Chakraborty, as I already mentioned, is working in Amity University, Kolkata right now, and he was the student of this very university. Then it was known as West Bengal University of Technology, and he is one of the first batch of MSc Bioinformatics of this university. So he obtained his PhD degree then from University of Calcutta, and he has done his postdoctoral research in the Department of Physical Chemistry, Indian Association for Cultivation of Science. He has earned lots of recognitions and awards and certificates. He is serving in different journals right now, and I'm not going to discuss about his publication because this is a long list actually. So far, more than 70 publications in his bag, and most of these are very impactful journal, peer-reviewed journal. He has some book chapters and conference presentations, seminars, workshop also. Now, most importantly, he has received some prestigious awards like Microsoft for AI for health, and the project was on probing the evolution of SARS-CoV2 spike protein during disease outbreak and its impact on human ACE2 recognition. He has got different other awards also. He is also acting as a reviewer of some prestigious journals like Molecular Biosystems, PLOS Computational Biology, and many more. He has research collaboration with not only India, but also in abroad. So with this, I am inviting Dr. Sundi Panchakabhati to start his talk today.
Okay, so hope I am clearly audible.
Yes, sir.
Okay, so good morning to you all. My sincere thanks and gratitude to Honorable VC sir for providing me the opportunity to share my work. I am also thankful to the HOD of the department, fellow colleagues, and special thanks to Dr. Chitta for coordinating my talk and help me out throughout the process. Thank you, Chitta. As you rightly point out, so this is kind of homecoming to me. And so I belong from the first batch of this university MSc Bioinformatics courses. And not only that, during my PhD tenure, I have spent a lot of years in WBUT. So I was working with Professor Joya Bandyopadhyay during the cell culture studies of my PhD. So I was strongly associated with the university, not as a student, as a researcher, and also in many times as a collaborator. Now I'm here my talk. Share my presentation. Is that presentation visible? Is that okay?
Yes, visible.
Okay. So today's talk is evolution of SARS-CoV2 during disease outbreak, applications of evolutionary bioinformatics and advanced computation. So
Please, Dr. Chakraborty, your sound is not clear.
Yes, sir. Very low, sir. So if you can be a bit louder, sir.
So now it is
Or you
okay?
can... Yes,
Yes.
yes, sir.
Okay. So as rightly pointed out, so I belong to Amity University, Kolkata, particularly in Amity Institute of Biotechnology. So this is our campus at Rajarhat Newtown, and this is our biotech department at the top. So this work we have been carried out during the last 2 years because this is a new coronavirus that emerged. In December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. So that rapidly spread across the world, and ultimately WHO declared it as a public health emergency of international concern on 30th January 2020, and a pandemic declared on 11th March 2020. So I'd like to discuss these dates. So our initial study is started, right? It is declared as an international concern, that is during February. So this is the latest data. Till now, we have 507 million of people are infected with the disease and a death of more than 6 million. So if you look that America, Southern America, Africa, Europe, Asia, India is highly affected during this disease outbreak. So the SARS-CoV2 is a new coronavirus, but coronavirus are not new. So they exist there are many coronavirus in human coronavirus, bat coronavirus, and other animal coronavirus. They primarily clubbed into 4 different groups: alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. So according to the taxonomic criteria, this SARS-CoV-2 belongs to the family Coronaviridae, subfamily Coronaviridae, order Nidovirales, genera Beta coronavirus. So this is the old nomenclature of SARS-CoV-2. That time it was called like nCoV. So now if you do a cross-sectioning of coronavirus, you will see this. You find an positive sense RNA-based genome that wrapped around a protein called nucleocapsid. There is a viral envelope, and within the envelope, you find primarily 3 structural proteins. The smaller one is the E protein. This red one is called the membrane protein. And this one is the spike protein that gives the corona-like structure. So that's why it is called coronavirus. And if you look at the genomic organization, so there are 6 open reading frames, WRF1A and WRF1B, that encode 16 non-structural proteins. So all these proteins are primarily responsible for viral replication. But as the structural protein are primarily encoded by the later half of the genome. You can find this is the S genome, S gene that encodes for spike protein. This is E protein, M protein, and these WRF proteins are responsible to evade the host immunity. If you look at it very clearly, you will find that in S gene there are 2 parts, S1 and S2, and there is a cleavage site at S1 and S2. At that point, in between S1 and S2. So there are many alternating hypotheses, and this is not particular or very specific to SARS coronavirus. That is, it is in general phenomenon for all coronavirus. So if this is a coronavirus, you have a spike protein, so it is recognized by the host cell receptor, and after this recognition, there is a conformation change. And what you find that these sites are now accessible by the host protein. So there are furin-like proteases, TMPRSS2, that cleaves this virus, leading to the cleavage of the spike protein, and there is the process of endocytosis of virus to the early endosomal system. And within that endosomal system, all the other content is tuned up, leading to the uncoating of the RNA gene. And you will see that it uses host replication machinery, transcription translation machinery to do the translation and replication. During this translation, it will form the NSP protein that form the RTC complex. That RTC complex ultimately doing the translation of this structural protein that leads to the formation of the virus, and ultimately the virus will mature in the RG complex and it will go to the process of exocytosis and ultimately leave the cell. So you can clearly understand. So there are a lot of intricate biology and details are associated with it, but you can get a point that it requires host, because host has this content of replication, transcription, translation machinery that initially support this virus to do the initial process of replication and translation. So host has a significant impact on the SARS-CoV-2 gene. Now, our work started, as I told you, during the early pandemic. So that time there is no variant, but it was very common that all SARS, like all RNA virus, SARS-CoV-2 has very high mutation rate and also rapid replicative time. So that leads to many mutations. And there are studies that reporting that SARS-CoV-2 has a high degree of adaptive potential. So our objective is to study the process of genetic diversification. Genetic diversification is a very dynamic process, and viral genome in different populations may under different selection traits. So that is very important. As I told you, that there is a role of host, and host immune system vary from in different backgrounds. So if you have a different ethnic background, you have a different immune response. So that therefore our question is that since that time virus is progressing from America to USA, from China to Europe, then to USA, then to Asia, so it kind of exposed to different types of ethnic background. So what is the effect of this ethnic background on the genetic diversification of the crop? So this is our first question that we are trying to address. So it is important to evaluate the selection pressure of different micropopulation of viral genome to understand the origin of genetic diversity. So that time there are very few genomes available, only 92. We manually check each and every genome. We found problem in 10 genomes with missing region and also ambiguous base representation. So ultimately work with 82 genes. And you can clearly see that SARS-CoV-2 is basically a 80-rich RNA virus, but there are considerable diversity in genomic content in each of the codon position. As at the first codon position, you have relatively higher amount of G.
Dr. Sandipan,
Yes.
Please, you louder, please, or you set your mic. Your sound is so low. I think for mic problem like this.
So can you?
It is now clear. It is now clear.
Yes. Okay. Okay. So I hope you heard my earlier part of the talk, right? Okay. So as you can see that this G on an average has a value of 19.59, but the first codon position guanosine is 25. So in a 80-rich genome, you have a higher prevalence of guanosine at the first position, and you will see that in relative codon usage index, there are some codon that preferentially use guanosine at the first position. Second and the third codon position are biased towards the AT content. Third codon position is particularly very less amount of C. So this is the codon adaptation, codon usage table of SARS-CoV-2. So this is true for SARS-CoV-2, but I am looking to tell you that I am asking to tell you that please look at the deviations on the data in 82 genomes. So these are a small set of data, 82 genomes only, but still in that data you have a considerable variations. So that means there is genetic diversification of the process. Now then we ask that, is there any pattern? So we really find the pattern. So there are few genomes that are evolving with patterns of mutation that are very uncommon or unlike to other sets of genomes. So if you calculate the rate of evolution, few genomes are evolving with a specific higher rate. These rates has a pattern. This is the disparity index. And if you calculate the selection pressure, that means it's a negative selection or purifying selection, and it's a positive selection, you will find that few of the genomes are evolving with a positive selection bias. So that means in that data, few genomes are evolving with positive selection bias and has a particular higher rate of mutation. So we reported the data. This is possibly first data that kind of a surveillance data which shows that there will be emergence of SARS-CoV-2 variants. So that published in Biomedical Research International. So that time, till that time, there is no variant emerged. So later we shift our focus to the spike protein, because we know that there are going to be many variant comes into the future, and spike is the protein that is the highly immunogenic or immunodominant protein. So therefore, the selection pressure will be highest on the spike protein. So if you look at the genetic or protein structural organization sequence-wise, so as I told you, this is the S1 domain, this is the S2 domain, and there is a cleavage site. So the receptor binding motif RBD exists in the S1 domain. So if you look at the structurally, this is the spike monomer and this is the RBD. So spike does not exist as a monomer. It's essentially a trimer where one of the RBD is in up conformation. So this up RBD is actually able to bind with the human S2 receptor and form this initial attachment complex. Now, if you calculate the binding affinity of this RBD with S2, you will find that SARS-CoV-2 binding affinity is 5 times higher than SARS-CoV, although there are only few variations of amino acid. So that means if there are few variations in the receptor binding sites, that may lead to higher transmissibility. So that leads to our next question, that is there any emerging variant with increased S2 binding affinity? So that time 4,000 genomes were available, and we analyzed this 4,000 genome, and we find that these are the mutation sites that spread over the entire spike protein. Clearly in the receptor binding motif or receptor binding domain, you have 5 sites with definite selection pressure, and these are those sites. So this is the entire RBD, And this entire RBD, this is the site which is called receptor binding motif, RBM, that directly interact with the AC2. So at the middle, there is an anti-parallel beta sheet and there is a ridge 1 and ridge 2. So this region entangles with the AC2 receptor. And you can clearly see that at this receptor binding motif, there are few population variant that already emerged during the early pandemic data. We then calculate the binding affinity. We find that two of the variant S494P and V367F, these two are with higher binding affinity. And if you find the reason, you will find that for V367F, there is an altered hydrogen bonding pattern where there is involvement of histidine 34 of AC2, and that involves the hydrogen bond with arginine 403. This altered hydrogen bonding pattern and formation of hydrogen bond network is possibly the reason of enhanced binding affinity. And for S494P, you took this region, you can see the arginine 393 of AC2 that involves in a long range of hydrogen bonding network involving tyrosine 41, sp355 to threonine 500. So there is a hydrogen bond network that is established in case of this variant. Although there are few losses of hydrogen bond, wild type hydrogen bond that also has been observed. So that time, few variants emerges, and we then explored these two variants, S494P and V367F with molecular dynamics simulation. So we calculate the distance between the S2 and spike, center of mass distance, and during our simulation, we find that both these mutant actually shows a reduced distance. That means spike is now able to interact with S2 with more prominent. So it reaches more closer to the S2. Now, if we calculate the number of hydrogen bond, then it is an interesting feature. You will find that for V367F, there is enhancement of S2 and RBD hydrogen bond. But for the other mutant, there is few losses of hydrogen bond. So that means you can explain the enhanced binding affinity of V367F in terms of forming higher number of hydrogen bond with the human S2 receptor. But what is the origin of enhanced binding affinity of this one? Because we can clearly see that there is a loss of hydrogen bonding contact. Few loss, though not many. Two of the hydrogen bonding contacts are lost. But you can clearly see from the distance that it approaches more closely to the human S2. So when you calculate the interaction energy, you will find that the van der Waals interaction energy is more favorable for this S494P variant. So that means it interacts with S2 more closely, and it can shows more surface complementarity with the receptor and forms the higher number of van der Waals contact. And that leads to the enhanced affinity of this S494P variant. So we report this data in BBRC, and that paper is highly cited in last year. So after we report this data, there are reports coming out that this variant SP494P become pandemic in northern UK, Colombia, Ireland, and later WHO declared it as a variant of concern. S494P is also included as a variant of concern. So we shared all our data with WHO throughout the process. So at that time, so we are at the end of 2020, and that time many variants come. So one of these popular variant, we know the UK variant, South African variant, Brazilian variant, Indian variant. So if you look at the UK variant and also South African variant and Brazilian variant, you have a mutation in 501Y. In addition, in these two variants, there is another mutation E484K and K417N. So these variants, so that time there is a concern, different reports are coming out. So that shows that these variants has an enhanced transmissibility. However, the vaccine efficacy should possibly will going to reduce, but there is a different type of opinion associated with all those type of variants. So we decided to start working on those variants. So this lineage one, so this is the UK variant, South African variant, and Brazilian variant contain an RBM mutation N501Y, which in vitro experiment shows that it has a higher H2 binding effect. But what is the effect of this mutation on antibody recognition? So that's a debate, because many paper shows many different contrasting data. So in a paper that has been shown that N501Y mutation shows a 10-fold decrease in efficacy during the neutralization of mRNA vaccine elicited MFs, but mouse adapted SARS-CoV2 N501 strain can be effectively neutralized by vaccine elicited serum. So there are contrasting reports. In addition, the Brazilian variant and also the South African variant, and there is also another variant of interest, P2, that reported in Rio de Janeiro and that rapidly spread in the northeast part of Brazil, that contains another spike mutation, E484K. So E484K, what is its efficacy in transmissibility is not understood, but clearly different data shows that it reduces the efficacy of vaccine. So we then try to understand what is the role of E484K and N501Y spike RBD mutation on ACE2 and neutralizing antibody recognition. So we do an extensive simulation, and our data clearly shows that wild type spike and S2 is going to stable after 100 nanoseconds of simulation. N501Y undergoes significant structural changes upon binding. You find 3 different structural conformational ensemble, but you will find that for E484K, there is a high stabilization of the complex. So at least you can understand that E484K mutation stabilize the spike S2 complex more in comparison to wild type. Now, if you understand the distribution of hydrogen bond in population, you can clearly find that there are 2 populations in wild type, one with a higher number of hydrogen bond, and this one with a very less amount of hydrogen bond. For N501Y, we'll find there are more population with considerable higher number of hydrogen bond, but there are significant population with low number of hydrogen bonds also. Likewise, for E484K, you have a considerable amount of population with higher number of hydrogen bond, also a significant number of hydrogen conformation with where the number of hydrogen bond is significantly reduced. We then do a cluster analysis and identify the solution structure, and you can clearly see that the binding interface, protein-protein binding interface, clearly changed in different populations. Now, if you can calculate the solvent accessible surface area, that means the area of spike that is exposed to solvent. So you can clearly see that there is a conformational population with higher solvent. So that means there is more solvent accessibility, less amount of protein-protein interaction sites. But for both the mutant, you can see that these high Sahasa conformations reduce significantly. So that means there is a population of conformation that shows high number of protein-protein contacts. Particularly, you will find that a region of RBD that is part of RBM, that is receptor binding motif, this region significantly stabilized in case of both the mutant. So this is the wild type data, and you can see for mutant N501Y, there is a reduction of RMSF. And particularly E484K, it is highly stabilized. We then calculate the binding affinity. You can clearly see that these 2 variants has a higher binding affinity with the H2. We calculate potential of mean force. That means the energy required to break a bound complex to make it free. So you can clearly see that more energy is required to break this spike S2 complex for these 2 variants, particularly the E484K variant. Now, if you want to look at the origin, we'll find the enhanced binding affinity is solely due to the pi cation pi stacking interaction and pi cation interaction for N501Y mutant. And for E484K, this is solely due to the effect of ionic interaction. So there are more ionic interaction in this case of E484K variant. So the enhanced affinity of E484K is due to enhanced ionic interaction and forming stable complex, and the enhanced binding affinity for N501Y is solely due to the enhanced number of pi-pi interaction and pi-cationic interaction. We then explored the effect of those mutations on the antibody recognition. So we consider the monoclonal antibody B38, and you will find and do an extensive simulation. You can clearly see that these two mutations changes the conformational ensemble in comparison to the wild type. And if you do a cluster analysis and identify the most stable solution complex, you can clearly see that there is a loss of contact for N501 mutant. Now we calculate the number of hydrogen bonds. So this is the heavy chain, this is the light chain. So if you calculate the number of hydrogen bonds between spike and H chain, you will see that there is a significant loss of hydrogen bond in the heavy chain. But in case of light chain, you will find that for N501Y, there is a reduction of hydrogen bond, but there is a little increase in hydrogen bond for E484K. So for N501K, N501Y, there is less number of hydrogen bond with both the H and L chain. But for E484K, there is significant reduction of hydrogen bond with the H chain, but there is a small gain in number of hydrogen bond with the L chain. When we calculate the binding affinity, we show that the binding affinity is clearly reduced in case of both the mutant. And if we find the origin, we'll see that there are several critical hydrogen bonds that are present in the wild type spike B38 complex that is absent in those variants. In addition, we'll see that for E484K mutant, there is a loss of pi-cationic interaction. So we attribute the loss of binding ability of this UK and South African variant to monoclonal antibody is primarily due to the loss of hydrogen bond, but for E484K, there is additional loss of pi-cationic interaction. And we published our data in International Immunopharmacology. So this is our proposed model. So possible pandemic variants are emerged with enhanced S2 receptor, and they reduce the binding affinity with the antibody. So therefore, it has an enhanced transmissibility, but it also has an immune invasion ability. So we then very interesting to know that where is this receptor usage comes from. So we ask a very critical question that what is the origin of this S2 receptor? Is this S2 receptor is unique to beta coronavirus? So that time there is a paper published in 2020 in Nature Microbiology. So you can understand that the paper published at 2020. So obviously the data includes possibly 3 to 5 years back. So this study does not include SARS-CoV2, but it includes SARS-CoV. So their data shows that these are all 29 beta coronavirus. This clade 1 beta coronavirus only capable of infecting humans with a differential ability of post-cell recognition or invasion. Clade 2 and clade 3 does not able to infect human host through the S2 receptor. Now you can then ask a question that, okay, so that means clade 1 beta coronavirus has this S2 receptor. So you will be amazed to know that it is not. So there are alpha coronavirus NL63 that uses AC2. Whereas there are other beta coronavirus like SARS-CoV, they use DPP4. So therefore, there is considerable amount of receptor tropism in beta coronavirus, and receptor usage does not follow the taxonomical classification. So we then ask a very critical question: where is this receptor usage comes from? Because this is infecting AC2 receptor looks like a critical event in host cell invasion. So how so we are asking this question that how this SARS-CoV2 gain the AC2 receptor usage, because it is not that it belongs to the beta coronavirus and all the beta coronavirus usage AC2. They have a considerable receptor tropism. Some use CCAM, some use sugar, some use AC2, some use DPP4 mono receptor. So therefore, there is considerable variation. And there is a data which shows that in case of this human infecting or human coronaviruses, they have a differential ability to recognize ACE2, but they does not bind with DPP4 on AP. So we took all these 29 beta coronaviruses. In addition, we take SARS-CoV2 also to understand where is this origin of ACE2 usage comes from. So initially we started with analyzing their genomic content, and clearly showed, you can clearly see that in genomic content distribution, SARS-CoV2 is an outlier compared to the other beta coronavirus. Even if you look the effective number of codon usage, you can clearly see that SARS-CoV2 is again an outlier in comparison to other coronavirus. And the most closest or prominent neighbor is this CoV ZX45 and CoV ZXC21. These are bad coronavirus. So we then ask a question, is there any selection pressure? So we do a parity plot analysis. We can clearly see that, okay, there is a selection pressure, because if it is evolving with sole neutrality, you will find that all these are distribution at the center of origin. But it is deviating from the center, so that signifies that there is a significant amount of selection pressure working on this beta coronavirus. So we then use the receptor, we then use the RSU-based principal component analysis. And here you also see a very interesting feature that all these human ACE2 user clubbed together, whereas SARS-CoV2 clubbed with two non-human ACE2 user bad coronavirus. So that gives, so genomic content analysis gives us the feeling that SARS-CoV2 evolution is possibly uncorrelated with the other human ACE2 user beta coronavirus. We then analyzed a very critical property, which is called the Codon adaptation index. And you will find a very interesting feature is that there is a critical mark of 0.7. Those coronavirus spike protein that has this Codon adaptation index of higher than 0.7, they are able to infect human host. So that means there is required of optimum codon usage that cannot be violated. So evolution must have to be worked maintaining this balance, that it cannot overrule the codon adaptation index. Critically, it cannot allow the mutation such that the codon adaptation index falls below the 0.7 limit, because these are non-human ACE2 users. So all ACE2 user more or less has this codon adaptation index of 0.7. So we then analyzed the different 29 beta coronaviruses based on different properties, and we find that SARS-CoV2 is significantly different from all 29 other CoVs based on relative evolutionary distance. But all the other CoVs are classified into two groups. This one is the ACE2 user, this one is the non-ACE2 user. And if you do a parity plot analysis, you can clearly see that SARS-CoV2 sequence variations is completely different than in comparison to 29 other beta coronavirus. And when we do a file, so we then analyze their sequence using multiple sequence analysis, and we find a very interesting feature that those coronavirus that are capable of infecting human host, they have these two insertions. However, those coronavirus that are incapable of infecting human host, there is two deletion regions. These regions are deleted in those beta coronavirus. So now once we do a phylogeny, we can clearly see that all ACE2 user human CoV, they belong to the same clade. But very interestingly, SARS-CoV2 does not belong to the ACE2 user clade. Rather, it belongs to a clade which belongs to the bad coronavirus incapable of infecting human host. And when we do a selection pressure analysis on the phylogeny, we'll find that positive selection exists on these clades, whereas positive selection is very little on these clades belong to SARS-CoV2. So that means these clades, receptor usage in these clades are driven by positive selection, whereas SARS-CoV-2 gained receptor usage through the process of recombination. So they are primitive bat-like virus that undergoes a spillover to in a intermediate host, where it gets recombined with these two regions and gets this AC2 receptor usage. So SARS-CoV-2 is not evolving through the standard receptor usage evident from other coronavirus. Rather, it evolves due to the process of recombination. So we then analyze that, okay, so SARS-CoV-2 is gaining receptor usage in terms of recombination, whereas other coronavirus gaining in terms of positive selection. So what is the state? So does these different types of selection pressure have a difference on the structure and dynamics of the spiked RBD domain? We clearly see that S2 binders RBD is more stable. So black one is the SARS-CoV-2, red one is the SARS-CoV. And this green one is another coronavirus that has human coronavirus that has the ability to infect human host. But all the other non-AC2 users that shows a lot of deviations from this group. So that means they are structurally very dynamic. And if you look at the origin of their structural deviation, you will find that these two regions are the region. So this is essentially insert one. This is essentially insert, sorry, this region is essentially insert one region, and this region is essentially insert two region. So that means in case of AC2 usage, the insert one and insert two is evolving in a way such that it stabilizes the spike RBD. We then calculate the binding affinity, and you can clearly see that SARS-CoV-2 has the highest binding affinity, followed by SARS-CoV, and this is followed by another coronavirus capable of infecting humans. Interestingly, this is a bat coronavirus that belongs to the same group or closely familiar with SARS-CoV-2, but that has a significant loss of binding affinity. And this is solely due to the absence of those two insert regions, insert 1 and insert 2. It is expected that non-AC2 user has a very little affinity in comparison to the AC2 user, and they shows very little binding affinity with the AC2. Now we then interested to identifying, okay, then what is the driving forces? So is there a formation of stable complex with the AC2 is the driving force, or any other factor? So we evaluated the RMSD, and we'll find that RMSD is not correlated with the binding affinity data. So you can see that non-AC2 user forms a stable complex with S2, whereas the binding affinity is very low. Whereas we'll find that SARS-CoV, where RMSD is little bit higher in comparison to other non-coronavirus, can able to bind with significant affinity with the AC2. So RMSD is not correlating with the binding affinity. Then we calculated the interfacial hydrogen bond. So that means the number of hydrogen bond involving spike and S2. Here we find a correlation. You will find that the SARS-CoV-2 form the highest number of hydrogen bond, followed by SARS-CoV, followed by another beta coronavirus that is capable of infecting human host. Non-AC2 user CoV shows very little number of interfacial hydrogen bond. So therefore, all these evolutionary forces are driving towards achieving the goal of optimizing interfacial hydrogen bond to gain the binding affinity. So there are different groups. One group is evolving with the positive selection bias. Another group is generated through a process of recombination, but towards the same goal that we have to achieve this optimum number of hydrogen bond between spike and S2. So this is the solution average structure. You can clearly see that this insert, so those coronavirus capable of infecting human host has this insert region that actually entangle with this AC2 and forms higher number of hydrogen bond. In case of non-S2 user, you can see the absence of this insert 2 region, particularly there is a significant loss in the contacts with the AC2. So we proposed the hypothesis that this is a spike S2 complex, and there are two different evolutionary forces are working. One group that belongs to SARS-CoV evolved with a positive selection bias. Another group which belongs to the SARS-CoV2 that evolves through the process of recombination. But the goal is very similar, that you have to enhance the spike is to bind hydrogen bonding contact such that your receptor binding affinity increases. But all these forces are working under the common edges that you need to maintain the codon adaptation intake index such that your translational efficiency does not fall. So because we saw that there is a limit of 0.7 codon adaptation. If it falls below, then the translational efficiency, it can infect the host cell, but it cannot replicate with too much of efficiency like this SARS-CoV2 is right now doing. So this is our proposition, and we proposed it in BBA molecular basis of the disease. It's currently under review. So we strongly acknowledge our students. So Sanjana, Trisha, they are worked on this project. Aditi, Chiranjit, and Rusha is currently working on different variants right now with me. And we, our lab is primarily funded by Microsoft. So we received two fundings from Microsoft AI for Health program. We received funding from DST SCRB, and recently we received funding from National Science Foundation USA for extreme supercomputing usage on this SARS-CoV-2 program. So this is only possible through their generous support of our funding agency. So not only the SARS-CoV-2, I am also involved in other projects. We work on GPCR activation mechanism in collaboration with Professor Amitabh Chattopadhyay. I have a strong interest in antiviral drug development. So Dr. Horacio Perez Sanchez is kindly helping us for AI part, and Dr. Ashley Brown and Namchul Ha, they are working for the experimental validation of our identified hits. I have works like possibly decades on Alzheimer's disease to understand the pathobiology and therapy and development. So this is my area of my PhD also, and I have spent a considerable amount of time in WBT working in this area. So thank you for your time and your patience and carefully listening to me. I'll be very happy to answer any question if you have. Thank you.
Thank you very much, Dr. Chakraborty. It is a very nice talk regarding this SARS-CoV-2, and interestingly, you have shown that applications of bioinformatics to analyze the data, so which will help the student very much. Among the audience, most of the students, and now the session is open for question answer. So if you have any question, you can directly ask to the speaker. Is there any question so far in the chat box?
I have
There are
few
two
questions.
questions in the QA box.
Okay. Yeah. So yes, Dr. Hiraranjan Boirago is asking some questions. Please.
Yeah, I have few questions to Dr. Chakraborty. You have run some MD simulation in SARS-CoV2 protein, ACE2 or RBD, and I would like to know what's the basic differences of MD simulation parameters of SARS-CoV and SARS-CoV2. Do you believe SARS-CoV2 is more dynamic in nature than SARS-CoV?
There is a paper published in possibly IGMS. Their claim is that in case of SARS-CoV-2 there is little bit more receptor dynamics during S2 recognition. So that paper has been published, but I have a small comment on this, that these I don't think that this is correct. I mean, I can specifically talk on this because the paper works on only the RBD. So, but you can understand that when it's binding to with the receptor AC2, that time it's the entire spike and it's a trimeric complex. So none of the paper has been considered the effect of those trimeric protein at that position. What is the receptor dynamics? But surely I can. So there is another paper published in JPCL that shows that for SARS-CoV2, as I showed up, so you need a RBD up conformation to bind with the AC2. So that dynamics is more evident in SARS-CoV2 in comparison to SARS. So therefore, you can say that even in trimeric form, RBD is possibly tends to up conformation more. So therefore, its ability to recognize S2 is higher. So that you can say at least.
But is there any specific residues at the both this site SARS-CoV or SARS-CoV2? Some specific residue is there that may be responsible for their this type of different behavior?
Dynamics is not, but for binding, I can say there are 3 to 4 residue at the binding interface that leads to 5 times difference in binding effect.
Okay. Thank you, Dr. Chakravarti.
Thank you.
All right. So we have another question from Anuj Bandral. He's asking that can we predict prospective variants based on mutation rate?
If you look at the mutation rate in my data, I possibly didn't show you. The rate of mutation is least in the RBD. For entire spike, the N-terminal domain and the C-terminal S2 domain is more prone to mutation compared to RBD. So therefore, actually spike is resistant to mutant in RBD. So if you want to classify this variant in terms of mutation rate, then you will end up with variants with more in N-terminal and C-terminal domain, not the RBD variant. So you cannot predict the RBD variants by considering the rate, because SARS-CoV2 is possibly maintaining the rigidity of RBD region. So if there is too much of mutation, the receptor usage is gone. So that's why possibly it's less susceptible. The rate is lowest in the RBD region in comparison to other N-terminal and C-terminal. So I think if you want to objective of you is want to identify the RBD mutant, then rate is not the criteria. But for N-terminal domain and C-terminal domain, this can be used.
All right. Next, Moumita Saha is asking that is there any online tool available for running molecular dynamics simulation after docking? So it is a more technical question.
Currently not, because simulation is a computer expensive process, very expensive resources. So online it cannot be handled. You require resources to carry out.
Yes. Then
but you can do a refinement. You can do refinement. There is possibly some web server available that you can refine your docked complex. So you can check with Rosetta or Haddock to define your initial dock complex, but you cannot get the simulation data out of it.
All right. Next, Devopriyo is asking what is the significance of Sahasa and B factor? Sahasa and B factor.
Okay, so Sahasa, as you can understand, that Sahasa means solvent accessible surface area. So if you consider the spike and AC2 complex, if your Sahasa is higher, so that means you have less amount of protein-protein surface available to interact with S2. So therefore, if you can check in the simulation that if your Sahasa is high, then that means your receptor interaction interface is low. And B factor is an experimental quantity that's related to crystallography. You can quantify B factor with the RMSF from the simulation. There is also equations available that can quantify the B factor from the RMSF data. So RMSF is a qualitative description of B factor. From the simulation, you can get the RMSF of individual residue that correlates with the B factor of your crystallographic data, and that gives the idea of stability of that.
Thank you, Dr. Chakravarti. Next, is there any more question? Okay, very few questions. Common questions are there, like how to control coronavirus. So actually, it is very difficult to say.
And it is very
how
difficult to say. So, or you can say that we have to obey the COVID guideline first.
Right.
Currently, this is the first and foremost process we have to maintain. We have to trust with the vaccine because currently no other therapy is there.
And another one, Ansika Das asked, will corona come again? Yes. So as he discussed, as our speaker also mentioned, the phylogenetic analysis of this proteins and all. So obviously this coronavirus is mutating and there is a high chance of getting this disease again, but obviously in different form, in different format. Am I correct, Dr. Chakraborty?
So coronavirus, there are coronavirus 400 years and 500 years back, and there will be coronavirus at 400 and 500 years.
All right.
And anytime it can jump over. There is receptor tropism. It's very common in this.
Right. Swapnil Pal is asking, sir, can we apply molecular dynamic simulation to know if SARS-CoV2 or SARS-CoV which have more mutation rate and why?
Molecular dynamic simulation is basically gives you an idea of conformational dynamics of the system. It does not give you the idea of evolution. It is not correlated with the evolutionary rate. So you have a fixed conformation and you only get an idea that how this conformation will going to change with time. So you can get an idea of stability. You can get an idea of binding affinity. You can get an idea of some parameter like hydrogen bond, solvent accessible surface area, but you cannot predict the evolutionary rate. With simulation, you cannot predict evolutionary.
Yes. Pritam Kumar Das is asking, is there any machine learning tool to calculate the evolution? Yeah, so that's a good question because I think Dr. Chakraborty is also learning the machine, using the machine learning techniques and all to his research also.
Yeah, but very few. Mostly I use computer simulation. So currently I am trying to, based on the simulation data, I currently want to devise an algorithm where we can predict future mutations.
Right.
So this is our current work, but we are not expert of machine learning. So we need help from our foreign collaborators to feed those data in a proper learning algorithm to get out more relevant information. But surely this is one of the hot topic of interest for computation biologists and chemists for new, possibly in last 2 years and possibly will be in next year.
Right. Okay. So Momita Saha again asking, can CapsFlex 2.0 and iMod servers are good enough to conclude about simulations?
No, these are not.
Okay. But she is saying that these two servers are well reported in many well-reviewed journals. So this is conflicting actually.
So what I suggest is that if you want to do simulation, so do simulation with proper taking care of parameter force field. And for that, there are several packages available like Gromax, Charm, and Ember. These are the more popular software or packages that are available for simulation. But do not trust web server data because this is not extensive simulation. You need to understand that to do a simulation, it's not only about a 10 or 15 nanosecond simulation. It's about a few hundreds of simulation to make your system equilibrated properly. Then you can study or analyze those data. So for this complex like RBD-S2, the size of this complex is huge. So at least it will take a few hundreds of nanoseconds to equilibrate. And I showed in my data that for wild type SARS-CoV2 spike and S2 protein that takes 100 nanoseconds to equilibrate. And later 400 nanoseconds, it forms the equilibrated structure. So you can analyze the data after running 100 nanosecond simulation. So that is the proper process of analyzing molecular dynamics data. So you have to ensure that your system is properly equilibrated then you can study and analyze those processes to get a meaningful information. Otherwise, you may be wrong.
Okay. So we are getting lots of questions. I think most of them cannot be addressed right now due to time constraint. Anyway, so I'm.
You have my email ID. If you find a relevant question, you can just
Sure. Sure. We'll circulate to the participants also. And one of them, Somodeep, is asking that one slide about an alternative hydrogen bond. Is it somewhat related to alternative catalytic mechanism, which is making the virus more hard to combat?
No. So this is nothing related to catalysis. It's absolutely binding. So molecular dynamic simulation cannot deal with catalysis. Catalysis is a process where there is bond formation and breaking. So molecular dynamic simulation in its structure is a molecular mechanics based force. So therefore your bond has to be made. So we cannot study catalysis using molecular dynamics simulation. It's only about interaction, receptor interaction.
Okay, so I think okay, Shweta Pal is asking, sir, if the nucleotide sequences can be altered in order to insertion, then can we control the binding affinity by altering the sequence changes?
Can you repeat it once again?
If the nucleotide sequences can be altered in order to insertion, then can we control the binding affinity by altering the sequence changes?
Yes, absolutely. And this is what we are doing right. We are trying to identify future variants. So what are those variants? So you can find, if you do a binding site analysis, and you can understand the contribution of each and every residue in binding free energy, you will find that there are few residues that are contribute more to the binding affinity with AC2. But there are few residues at the binding surface that does not contribute significantly. So a possible route of evolution is that residue is going to mutate in a way such that it can gain more binding. So we are actually interested in identification of future variants that can mutate in such a way that it can mutate that those residues that are currently not contributing to the binding affinity, that they are now become contributor to the binding. So we are currently right now we are actually doing that. We are actually interested in the insert 1 and insert 2 region, and we are trying to optimize that what is the optimized residue that gives the highest binding affinity with AC2. So this is absolutely possible, and this is the current working area of me also. So what is the future variant, possible variant? I'm not telling that that will appear, but what I'll say that if that appear, that has a higher rate of binding affinity with H2. Therefore, it can relate with the higher transmissibility.
Sir, please unmute, sir.
Yes. Okay, so I think there are a few more questions. One is asking, can the vaccines be able to stop the evolution of COVID in the human body?
This is another very interesting question. I didn't show that data. What vaccine does is that vaccine modulates the evolutionary path of SARS-CoV2. So as you can rightly see that currently throughout the years of million years, the evolution is directed by selection. So SARS-CoV evolution is driven by selection. SARS-CoV2 evolution is driven by recombination. But now SARS-CoV2 fails a vaccine. That means it is now challenged by our immune system. And we see that the variants that are emerging is absolutely as a result of vaccination. So if you point out those variant mutations, you will find that are the critical residues involved in antibodies. So currently, the selection process of SARS-CoV-2 is basically they want to evade the immunity. That's the driving force. So basically, again, the fact is that this is a very short time span data. So if you do an evolutionary analysis, it is better to do it in a time frame of 500 years or 400 years. Then you have a pattern. But for the very short time frame, there are fuzzy data. So that is very difficult to tell that what is this effect of 2 years on the entire evolution of SARS-CoV-2. But if we solely consider the time period for this evolutionary analysis in this 2 year, we see that the current driving forces is basically evading the vaccine elicited antibody. That's the driving forces, and the variants will appear due to these.
All right. So I think the students got their answer. So with this, we should stop here. And if you have any more questions, you can share with me, or you can directly share to the speaker. We can share the email ID of him, and the link for feedback has been shared, so you can give the feedback in the chat box. It is given. You can find. So now it's the time to give thanks again. First of all, I must thank Dr. Chakraborty again to come and share his views on his current research. We hope that in future we can collaborate with him with some bioinformatics project, and when we are in offline mode, we will invite him to our campus again, also in the new campus, not his old campus, in the Horinghata campus, and hopefully he will come and will enlighten us with his insightful talk. Next, we also very much thankful for towards the VC sir, who gave us the opportunity to conduct such kind of webinar in online mode. So, and we also thank to the IT department of McLeod University, especially to the IT team and Mr. Prem Kumar. From the very beginning, he is instrumental to design this fire and to organize this thing smoothly. Thank you all. And at the last but not the least, all the audience and the panelists also for giving the time, valuable time. So with this note, I'm ending this session today. Have a great day.
My sincere thanks to all the panel members and also VC sir, and especially to you, Dr. Chittabrata. So giving me the opportunity to share my views, and also I'm very interested and delighted to see that after 1.5 years, still more than 350 participants are there. So I hope they are find interest in the talk also. So we'll be very happy to collaborate with you. So you know all this, we have talked regarding this in different times. And also anyone, so if you find interest in my work, so please contact me. So I have my email IDs available with Ciptosat. So kindly feel free to contact me, and if I can be of any use, I'll be very glad.
Sure.